Okay, perfect. Okay, so let's go ahead and get started. Thank you everyone for joining us for this Amy seminar today. We have Hairong Wang, who's a PhD student at Georgia Tech. She works on machine learning models in clinical settings. And interestingly, she is graduating and will be an assistant professor at UT Austin this coming semester. So very much congratulations for that. And I think we're in for a very excited talk. So please take it away, Hairong. Thank you for being here.
Yeah. Thank you so much for the very kind introduction. And hi, everyone. My name is Hairong. I'm currently a final year PhD candidate at School of Industrial and System Engineering at Georgia Tech. And I'm very fortunate to have the opportunity to be here today to share my recent work on advancing precision medicine, knowledge-informed methods with data and computational efficiency. So a little bit about myself. So my research mainly focuses on two main research directions. One is data efficient AI models for healthcare, and the second one is optimization for AI in healthcare. So I will focus on the first one in today's presentation. I'm currently a student at Georgia Tech, and I obtained my bachelor degree in math from University of Oxford. All right. Let me start for today's presentation now. So when we talk about the precision medicine, we all probably roughly have our own understanding. Just generally speaking, the customized healthcare using genetics, physiology, and lifestyle mainly will identify it as the precision medicine. And the importance of the precision medicine have been also widely acknowledged. First of all, the most important thing that we care most is the clinical outcomes, right? So based on the published study, if we apply the precision medicine to the treatment, then the clinical outcome can be improved largely. And in addition, from a financial perspective, so the precision medicine can also bring the great financial return based on the report from the PwC recently. And then due to these great benefits of the precision medicine, it has already become a national strategy. Actually, the precision medicine has been written into the 21st century course action, which is mentioned which is the US law, and in total around 1.45 billion has been invested to the precision medicine initiatives to support the research related to this topic. So clearly the precision medicine is very important and can affect everyone's life. Then how the precision medicine progressed over the several decades. So historically, the precision medicine mainly focuses on the one treatment fits all model, which means the treatment is to only have a low fidelity precision. So one typical example is the TB here. So the standard treatment can be applied to the patients in different groups. And however, as the time goes by, the alternative goal of the precision medicine have been improved to the one treatment per group or even one treatment per patient. So one example here is the breast cancer. So, if we want to treat the breast cancer, then based on the biomarker, the test results of the biomarker from the patients, different type of therapy, including the targeted therapy or hormone therapy, will be applied. However, there are still a type of disease that need even higher fidelity precision medicine. For example, the cancer treatment. because cancer has heterogeneity inside each entire tumor. So which means if we can develop the localized treatment to each small specific subregion, then the treatment will be more effective. However, very unfortunately, no satisfactory solutions to date has been developed. So today, I'm going to share some recent development for bringing us closer to the high fidelity precision medicine. So before diving into the model, I would like to provide some context about the glioblastoma. So when we consider glioblastoma, which is one of the most common and aggressive type of brain cancer, and the median time to death is only 14 months. And what even more worse is that glioblastoma has an extremely high recurrence rate. So around 90% patients experience recurrence after the first round or primary treatment, including the surgery, radiation, and so on. And the median time to recurrence is only 6 months. But you may wondering why the recurrence rate is so high and why the current treatment fails because the mean time to death is only 14 months. So actually, we will mention just like we mentioned in the in the introduction, all tumors exhibit heterogeneity across different regions, and it turns out that all methods neglect this kind of heterogeneity. And as a matter of fact, the tumor can exhibit different types of heterogeneity in different aspects. For example, the genetic heterogeneity, the immune environment can also exhibit the differences in the immune cells composition and the responses. And for the cellular differentiation, different levels of differentiation can also affect the treatment outcome. And finally, the proliferation heterogeneity, which refers to the variability in the rate of tumor cell growth. Therefore, we know that heterogeneity is one of the main reasons that leads to the different treatment response across tumor regions. So like shown, we can take a look to this figure together. So if inside each tumor, for each small subregion, some subregion can be regarded as the benign, some subregion will be grow very fast, and some of the region can would be identified as the immune cells. Then if we apply exactly the same treatment to all these different subregions, then which means some subarea will be overtreated, but some area will be undertreated. So that's also the main reason why why the current treatments fails. So okay, so clearly we need the development of precision medicine. And our next question is how can we achieve it? So actually there are two essential demands. The first one, like we have already seen is the fine-grained characterization of tumor heterogeneity, because this kind of information can be very helpful to guide the future localized treatment design. And for the second one, go without saying, ensuring the patient safety is also very important. So let me take an example to illustrate these 2 points. So SOX2, which is a gene to indicate cancer stem cells. So if we are, suppose we are considering SOX2 expression, and for any given region, suppose we can accurately get the information about how high about the SOX2 expression. For example, whether it is under the normal level or whether it is overexpressed. Then we can design the treatment by the very precise treatment plan, including the CAR-T therapy and some other very typical therapies. Different types of therapies can be involved together. But very unfortunately is that this kind of information, the gene expression, cannot be measured without invasiveness. So which means just like shown here. So if we want to get the detailed measurement for the gene expressions, we have to take the small piece of the fresh tumor tissues out. But maybe you have already known that this kind of biopsy is invasive, so it's not feasible for the whole tumor diagnosis. And to lower the risk of the patient, only very small number of biopsies will be taken from each tumor. So our next question is, what can we do, or any information that is safe? What kind of information that is non-invasive we can leverage? So I believe many of you know about the MRI, like shown here. So MRI offers very rich and detailed information that is critical for effective treatment planning. So if we can produce a fine-grained expression level prediction based on the MRI, and very few biopsy or even no biopsy are needed during this process, then that would be great. That would be ideal, right? However, currently, there are no existing work that is capable to achieve this outcome. So today I'm going to present my recent research that combines the data science and operations research to achieve this kind of new capability. So of course, this is by no means an easy task. And I would also want to argue that this is actually a very challenging task. So actually there are three major challenges. So the first one is the very limited labeled samples, because just like we mentioned, because of the invasiveness of the biopsy, there are only very few biopsies can be taken per tumor or per patient. So in general, there are only less than 3 biopsies will be taken per tumor. So in this case, the supervised learning method are not reliable because our labeled data is the sample size is very, very small. And then the second challenge is the cross-region interdependencies. So like shown in this figure, so there are very complex microenvironment interactions, and based on the current medical knowledge, the precise mathematical model to describe this kind of complex microenvironment interactions are unknown. So it is unclear for us to know how to harvest this kind of interactions and how to extract this kind of interactions information to guide the machine learning model learning. And lastly, the third challenge is about the computational efficiency. So in most cases, we have to train a very large and complex model with some kind of constraints, some linear constraints, or some even non-linear constraints. So the novel algorithm or novel optimization algorithms are required to improve the computational efficiency. A set of tools and methodologies has been developed based on the three main challenges. So in summary, my ongoing research has been driven by these three aspects. And I have developed new tools and methodologies from data science to operations research. So specifically for the first challenges, I'm going to introduce a semi-supervised learning model to mitigate the sparse label data. And for the second challenge, I will introduce a data-driven model that can capture the regional independencies from the global context. And these two are the main focus of today. And in another direction of my research are focusing on developing the optimization tools which can use to help improve the computational efficiency of the AI for healthcare model training. So by combining these three developed tools, my research helps enhance the high fidelity precision medicine. And very fortunately, I have the opportunity to collaborate with different medical institutes during my PhD study. And in the future, I also want to further leverage the developed method to other different kinds of disease, like shown here. So now in this talk, I'm going to discuss two representative work for addressing this challenge mentioned in the previous slide. So first part, I'm going to introduce the knowledge-informed framework for recurrent glioblastoma. And for the second part, I would like to introduce the data-driven modeling for global dependencies in glioblastoma. Okay, then let's dive into the first topic. So when we are talking about the treatment of recurrent glioblastoma, we are wondering what we can do. So actually, there are two types or two groups of tissue-specific gene models that controls the status of the glioblastoma, which are shown here. The first one calls the REACT model, which is with a high correlation with biomarker CD68. So if the enrichment score of a REACT model is very high, which means the proportion in this subregion, the proportion of the immune tissue is relatively high. And another one, another key gene module is recur module, which is with a high correlation with biomarkers SOX2, KICT8, and total cell density. So from here, I believe you have a rough understanding that if the enrichment score of the recur model is relatively high, which means the proportion inside each subregion, small region, the proportion of the recurrent tumoral cells is very high. Therefore, to identify recur and react enrichment scores, that is very important for the localized treatment design for the recurrent glioblastoma. Unfortunately, this kind of measurements of recur and react enrichment score requires biopsy, just like we mentioned in the introduction. And because it is invasive, so in general, only 1 to 3 biopsy samples will be taken for each patient. So what we can do in this case? What kind of other information do we have? So like mentioned, we have the MRI information. And MRI information is non-invasive, which means very safe to the patients. And at the same time, it can cover the whole brain, which means around for each whole brain, around 10 to power of 5 subregions. The small regions can be covered inside this the whole brain. But because there is non-invasive, so we cannot get the direct measurement of the module enrichment scores. So given MRI, our interest is to predict for any region, any small region, its enrichment score with the guidance of the very limited biopsy samples, so just like shown here. So actually it's a challenging task. Maybe you can imagine. As a matter of fact, here we can see a result from an existing approach here. So based on the random forest, the prediction of the enrichment score for each small region will be very similar to each other, which means the model do not have the capability to identify the specific enrichment score for different gene modules and different subregions. So why do all the current existing methods fail? So first, the main reason is the limited label data, because if the current model is focusing on the supervised learning method, then it cannot work. And the second main reason of failure is that the current image-based feature for MRI is also not ideal, as it is hand-designed. Because for the current existing method, they are focusing on use the texture features that are extracted from the MRI instead of directly use MRI. And unfortunately, existing study focus on the purely supervised or generic semi-supervised learning. So, which means consequently, they fell short of handling these challenges. Now, to tackle this challenge, I would like to introduce the knowledge integration-based weekly supervised learning. And in this process, we also construct a novel auxiliary task that can help us model the medical knowledge and helps learn a much improved feature representation. So let me introduce the auxiliary task we consider first. So for this one, the neural model, which is closely related to the target model, recur and react. For the neural model, which can be understanding in the same way as recur and react, it has a very high correlation with the biomarker neural. So if the neural model enrichment score is very high, which means in this small subregion, the proportion of the neural cells is very high. So can be regarded as the benign tissue. So you may wondering, why do we care about the neural model? So actually, a particular benefit of the neural model is that we have a biological relationship between the neural model and recur and react model that we can utilize to introduce the knowledge to the model training. So especially so like shown in these two formulas. So if the neural is very high, which means in each subregion, we do not have much room for recur and react. So in this case, recur and react enrichment score would be both very low. But if neural score is very low, which means there are much room for recur and react, then in this case, recur and react follows a negative correlation relationship. They will compete each other to get the room inside this subregion. Unfortunately, neural score, neural as the gene module, also requires biopsy. So what we can do now? It's actually possible to obtain abundant the specialized inspected mask, which can serve as the weak labels of neural. So let us take a look to this figure together. So we can see the small red area, which can be regarded as the tumor core. So all the samples inside this area will be regarded as the neural score very low, very small. And this blue circle, which is manually notated by the doctors. So if the samples fall outside this very big RY, the region, then the samples can be regarded as healthy, which means the neural score for the samples outside this blue circle can be identified as has a neural score very high. All right, then so we can see from this knowledge design, there is some kind of hierarchical relationship between neural and recur and react, right? So motivated or inspired by this kind of hierarchical relationship between these three gene models, we designed the bottleneck model with bi-level framework. So before start, I would like to mention what we will do naturally do if without this kind of knowledge guidance. So generally, if we focus on the supervised learning, we will just use the label data and train a model by using the loss function focus on the prediction result, right? However, if we design this kind of auxiliary task, neural, and leverage the knowledge information, then we can design the hierarchy framework in this way. So first, we have the MRI, which can provide abandoned unlabeled samples. Then we will train a model for only neural scores. And if the model can perform very well, which means we can use the model to predict the neural score for all the very abundant and labeled samples, right? And according to the status of the neural for each unlabeled sample, for example, if the neural is with low score and whether the sample with a high neural score. Then according to this information, we can specifically tell the lower level tower, the model, about which kind of relationship between the recur and the react should follow based on the given neural status. Great. And this is a general idea about the framework design, and this is what we have. So first, we have the rough neural label from MRI and mask. We also have the conditional medical relationship from here. So we regard this relationship as knowledge that we have. So how do we utilize these two data sources together? So we can now introduce the weakly supervised bilevel learning framework that can fully utilize these two information. So it has a bilevel design. In the upper level, our goal is to solve the auxiliary task of predicting neural status. And two things can be obtained by using the auxiliary task focused model. The first one is the neural predictor. So based on the neural predictor, we can get all the predicted neural status for all the unlabeled samples. And the second one is the feature embedding. Then this one can provide us more task-specific features that can learn by the auxiliary, guided by the auxiliary task. We will then use the neural predictor to encode the knowledge. And together with the improved feature embedding, we will proceed to the lower level, which solve the targeted task of predicting models of interest. So our lower level focus on the recur and the react prediction, but at the same time, it is enforced to follow the biomedical relationship of the 3G modules. So by using this generic framework, we propose BioNet. So for the upper level, the goal of the upper level is to predict whether the region of interest, or for each very small subregion, whether the region is with high neural or low neural. And the process is shown like this. So first, we initiate a pre-training step, which is focused on using the abundant weak labels, just like shown here, by using the mask that manually drawn by the doctors. And after that, we get a pre-trained model, and then we fine-tune the model by using the biopsy samples with very precise neural enrichment scores. Then in this case, in this way, the performance of the model will be further improved. And now we are ready to solve our target task of interest in the lower level. So we have two data sources. One is the label data, which is taken from the biopsies. And another one is the unlabeled data, which also have the predicted neural score, which is predicted by the previous upper level model. So our network has multitask learning structure with shared embedding in the middle. like shown here, and the aim to promote the information sharing between the different tasks, neural prediction and the recur prediction and react prediction. And specifically, we have two towers for our target models, recur and react. And now, very importantly, we will actually have another tower, like shown here, trying to predict neural. So you may wondering, why do we need to do this if we already know the status of neural? Well, this is because by learning the neural model, we will extract relative information encoded in the shared embedding here, which can help the prediction of the power of the other two models. And loss function, the overall loss function can be regarded as two parts. Is the first part is the prediction loss based on the three enrichment scores of the three G modules, and another one is the loss function designed for knowledge. So I will introduce the design for knowledge in details. So we first start with the generic knowledge that recur and react cannot be both high. So for the first term, we will use this term to enforce the model that cannot predict recur and react to be both high if the neural status is very high. Then we will move to the second type of the knowledge, which is recur and react should be low in high neural region. And then the third part is if the neural status is very low, then recur and react should follow negative correlation relationships. So okay, that concludes the main design of the BioNet. Now we are ready to apply this to an application of the recurrent glioblastoma by using real collected data. So this data, very fortunate to collaborate with Columbia University Medical Center. So they collected the data which including 69 biopsy samples from 31 patients. And at the same time, around 10 to power of 5 unlabeled samples are leveraged in the model training. As you can see, the prediction of the enrichment score for recur and react have some very appealing features. First, they have a high precision, which should be consistent with the biopsy. And it's also high contrast, meaning that it is truly reflect tumor heterogeneity. And for the prediction map, it's also consistent with the medical common knowledge. It's also required to consist with the medical common knowledge. And here I want to highlight that due to these nice features, the BioNet's prediction provides very helpful information for the follow-up targeted treatment or diagnosis. For example, the doctor can use the prediction to decide which small area should the next biopsy be located for further diagnosis. We also compared the BioNet with existing state-of-art methods. So we categorize the existing method into supervised and unsupervised model and compared with the best of each category, like shown here. So as you can see, this prediction does not have any of the appealing properties associated with the BioNet. Consequently, their prediction will be uninformative and unhelpful for downstream treatment. So when we look at the superior performance of BioNet, the ultimate reason is that unlike supervised and semi-supervised model, it can use a labeled sample in a way that encodes specific medical domain knowledge into the learning process. Sorry. And this type of capability is very new in the literature for AI for healthcare. So that's also the main reason why the BioNet can significantly outperform compared to the existing state-of-art methods. And as we can see here, when considering classification of gene expression level, the BioNet also substantially improve over existing method. So this shows the power of BioNet is quite general, has a quite good generalizability. So in summary, we have introduced the structure of a salary task with weekly supervised samples. And we also mentioned one improved feature learning here. And in its design, we use some novel elements, including the integration of the structured medical domain knowledge and the feature learning encoding. And we demonstrate that due to this all these novel elements, the predictive power of BioNet is much better than existing generic machine learning models. Okay, and that's the first part for today's presentation. I would like to pause here and to know whether there's any question for now for the first work. No? Okay. Then we are we are going to see one can the second one by harvesting the medical knowledge that encoded in microenvironment interactions. So let us move to the second one, data-driven modeling for global dependencies in glioblastoma. So before start, when we see the microenvironment interactions, you may wondering, what is microenvironment interactions and why do we need to care about it? Well, this generally refer to the complex interdependencies across tumor regions. For example, we have different diffusion across tumor cells. We have propagation of immune suppression mechanisms and diffusion of tumor effects. So a key observation from these complex interactions is that tumor status across regions are actually independent. So what does all this mean? Why do we need to care about this kind of interaction? Well, above all, this implies that using local feature for biomarker prediction, such as the gene models, just like we mentioned in the previous study, is not enough, because this is indeed all existing methods are trying to do. And more specifically, for this type of local methods, given an MRI local region, it will map it into local feature and do prediction based on the local feature. Now, the reason why local feature paradigm does not work is precisely because it completely ignored the interactions. We can have a look to this figure together, which means it's clearly not desirable given our observation above, because they are dependent with each other. If we want to predict the enrichment scores for each subregion, and we only use the localized feature but ignore all the surrounding features, then we will lose a lot of helpful information. So instead, local features, imagine we have certain what we call the contextual features. So like shown in this figure, more specifically for any local region X, like shown here, we will simultaneously consider all its surrounding regions, like all the Xi here, which we named the global context C, and construct the contextual features as a nonlinear function of X and its context. And clearly, by carefully designing nonlinear function phi here, the nonlinear function phi here, we can capture the interactions of X and its context, Xi. Consequently, if we use the contextual features as the base for biomarker prediction, the prediction will now utilize both the local information and, more importantly, taking into account of the local global interactions. But clearly, it's not an easy task, right? This is because macro interactions are actually difficult to model. For instance, unlike the approach we take in BioNet for complex logical relation, it is difficult to design explicitly regularization to enforce such relationships. On the other hand, explicit construction of function phi based on prior knowledge is difficult, and we have to use a data-driven approach. And at the same time, since there are thousands, if not more, macro environments or macro interactions, identifying all these kind of relative ones for the specific biomarkers is gonna be very difficult. And finally, this is our old friend that we only have very limited number of samples for biomarker prediction. So to tackle these fundamental challenges, we will first propose to capture local global macro interactions through self-attention mechanism. This will help us construct a generic contextual feature. Then we will construct a relative auxiliary task to help further guide the contextual feature to capture information related to our targeted task, which is the biomarker prediction. And finally, we will use the pre-training and fine-tuning framework to deal with the issues of limited sample from targeted task. So in order to introduce our full learning framework, let me first introduce the self-attention mechanism in the text of our biomarker prediction. So for a given tumor MRI scan, we are going to first split the whole MRI to different small patches. Then for every patch, we are going to assign 3 embedding vectors. It's named query, key, and value embedding. They will essentially serve as a representation of these patches. Now, for any given patch, we are going to identify its context, that is, all the preceding patches and succeeding patches, like shown in the small blue box. And we are going to produce the interactions of the current patch, which is context by using their embedding vectors into a nonlinear function called attention. And the attention function intuitively is very simple. It's gonna first look at the similarity of the current patch and its context through the inner product between their query and key embeddings. Then the attention function will simply aggregate all the value embeddings according to the similarity scores. As we move to the next patch, it can capture the global context from these kind of interactions. And in fact, it has been shown that composition of multiple attention layers can indeed can indeed capture the complex sequence-to-sequence function. Attention will be useful for capturing macro environment interactions. So this is the reason why attention is so crucial in constructing our textual features. Now with the attention mechanism in place, we are ready to describe the general framework of the proposed method. We call it SSMAE model. Specifically, we will start with MRI images, which is very safe and non-invasive information we have. And we will use the attention-based neural net called encoder to map the image into its contextual features. And then use the decoder to try to reconstruct the image from the contextual features. And the underlying idea is that we will try to learn a contextual feature phi that contains enough information so that we can actually reconstruct the original MRI. Now with the generic contextual feature in place, we are going to further fine-tune it by building another auxiliary task, which is the segmentation. More specifically, we are going to use a contextual feature to learn a tumor segmentation task. Learning tumor segmentation is actually very useful because it closely related to the tumor biomarker prediction. And moreover, we can actually readily obtain a large amount of label data from existing data sources. And finally, by learning the segmentation task, we can now make our contextual feature more relevant to the biomarker prediction. And now we are ready to tackle the main task of interest, the biomarker prediction. Specifically, the loose, we will feel so will feature a standard prediction loss. So the model training and the loss function will feature a standard prediction loss. And moreover, we will design a physics-based regularization such as the symmetry and smoothness of the prediction to further help the regularize the learning process and encode the necessary physical knowledge. So that's the basic high-level idea of SSME. As we can see, each of the auxiliary tasks is essential for building a contextual feature that is relevant to our targeted task. And at the same time, trying to capture interactions among tumor regions. And the power of the SSME model has also been demonstrated during our collaboration with Mayo Clinic. The task of interest is to predict the expression level of EGFR, which is the key gene for the targeted therapy. And as we can see here, thanks to the usage of meaningful contextual embedding, SSME model produces results that is simply unmatched by existing methods that rely on local features. So I would also highlight that contextual embedding can also be used to generate patchwise prediction mapping for EGFR or other for the entire brain or some other related genes, similar to what we have seen for the first product for the BioNet model. So great, we have seen a BioNet that utilized the explicit domain knowledge to enhance the prediction. And we have now seen how to create a contextual embedding to harvest the macro interactions of tumor cells and the better predictive power. And that's the summary of the second part. And before start to conclude today's talk, I would like to take this one last moment to thank these wonderful collaborators that I have for the past few years. And thank you very much.
Great. Thank you, Hairong, for such an interesting talk. Let's give her a virtual round of applause.